Welcome and thank you for joining this webinar on the efficacy of Dupilumab and relevant therapeutics in the immune atopic dermatitis model. In this session, I will walk through the biological rationale for modeling atopic dermatitis, the way the immune AD model is characterized, and how the model responds to selected therapeutics including Dupilumab, abrocitinib, IL-13 directed antibodies, and topical corticosteroid interventions. The aim today is to show the reasoning behind our model, the readouts used to assess it, and the type of questions it can answer. So we move on first to the structure of the webinar. Today's webinar is divided into 3 parts. I'd first like to introduce atopic dermatitis as a dynamic and heterogeneous immune disease. That includes a brief discussion of timelines, immune endotypes, serum and skin biomarkers, and the therapeutic landscape. Secondly, I will move into the immune AD model itself. We will look at what the baseline model looks like and how we quantify induction, as well as how relevant interventions behave in the model. Finally, I will finish with how an investigation can be planned, including pilot work, donor panel expansion, and selection of readouts. The key point to keep in mind from the start is that AD is not a single fixed biological state. The modeling challenge follows from that. Atopic dermatitis is often discussed through the lens of type 2 inflammation and that it is absolutely central to the disease. But AD is also dynamic, heterogeneous, and dependent on disease stage, patient background, and tissue context. In early disease, we often see strong epithelial alarm signaling including TSLP, IL-33, and IL-25, together with dendritic cell activation and Th2 skewing. As the disease becomes more chronic, type 2 inflammation persists, particularly IL-13, but the inflammatory environment can broaden. Th22, Th1, and in some patient groups, Th17 associated pathways can also contribute. These can be defined as endotypes, and endotypes can shift over time within patients. Additionally, the severity of AD can increase or remain lower over time, and is accompanied first by erythema, mild spongiosis, and limited immune cell infiltration. As the severity progresses, spongiosis grade and immune infiltration increases, along with a general increase in the milieu of cytokines. Itch tends to become more pronounced, and epidermal thickening becomes noticeable. At its most severe, the skin is thickened, lichenified, and may be perforated and bleed due to scratching and irritation, leading to a strong impact on quality of life. So chronic AD is not simply less Th2 or more Th2. It is a broader and more complex tissue inflammatory state. That is where the modeling issue begins. If the disease is dynamic and heterogeneous, then a useful model needs to capture more than one isolated pathway. It needs sufficient human relevance and sufficient biological complexity to detect meaningful therapeutic modulation. So this same complexity is reflected when we look at the biomarkers. Serum biomarkers provide one way of tracking disease-associated immune activity. A very recent human study published in the journal EAACI, dated 31st of July, puts these marks front and center. Several of the markers shown here are strongly associated with AD severity. CCL17, also known as TARC, and CCL22, or MDC, are linked to Th2 cell recruitment. IL-13 is a central type 2 cytokine, and MCP4 is involved in eosinophil and monocyte recruitment, while IL-18 reflects innate inflammatory activity. The table also includes markers such as MCP3, CD137, IL-17C, and CXCL9 and 10, which reflect a broader immune picture, particularly in chronic or severe disease. Point one here is that no single biomarker defines AD on its own. Rather, serum biomarker patterns show that AD severity and phenotype can be followed through measurable immune signals. And point two here is that we need to define clearly what markers should be measured and what not in a model. From our data, as we will show later, IL-13, but in fact not IL-4, is highly measurable within an explant model. Similarly, TARC, and by extension MDC, and MCP4, which tend to be tightly correlated, is a highly measurable supernatant marker. And the serum is only one layer. The tissue itself gives us a very different and important view. The skin biomarkers show us how the inflammatory phenotype changes within the affected tissue. In weaker or milder AD, TSLP might be present, but variable, and type 2 inflammation may be detectable. Some biomarkers of barrier function, such as filaggrin, loricrin, and involucrin, may be partially retained. Epidermal thickening and spongiosis are also measurable by histopathology. In stronger or more severe AD, TSLP tends to be more pronounced. Type 2 markers such as IL-13, again associated signatures, increase. Chronic epidermal activation becomes more evident, and barrier and differentiation markers such as filaggrin, loricrin, and involucrin, as well as tight junction components, can become reduced or dysregulated. Combined with the soluble markers, we have a clear biomarker panel for readouts, allowing therapeutic modeling to be interpreted in tissue context as well as through soluble readouts. And that brings us to the therapeutics landscape. The current landscape is broad and mechanistically diverse. There are physical interventions in the form of emollients, moisturizers, and barrier repair topicals containing active compounds as a first-line therapy against mild to moderate AD. Then there are biologics that target IL-4 and IL-13, for example, including Dupilumab through IL-4 receptor alpha, and others that more selectively target IL-13. There are JAK inhibitors, such as abrocitinib, which act more broadly across cytokine signaling pathways. And there are topical anti-inflammatory treatments, including corticosteroids and nemolizumab, which inhibits itch by blocking IL-31 receptors on nerves local to the skin region. Emergent therapies aim to improve on delivery and dosing and hit new targets such as the OX40 pathway, inhibit TSLP signaling, or as multispecific antibodies against a combination of these markers. This means that a useful AD system should not only generate an AD-like phenotype, but it should also allow us to ask questions about whether the interventions with different mechanisms can modulate those phenotypes in useful ways. Ideally, a model will be able to switch between some of these different systems. And that brings us to the practical hurdles in modeling AD. Firstly, atopic dermatitis is a heterogeneous disease, as we've discussed. Clinical translatability is difficult. Buyers need a human-relevant model, and models need to detect therapeutic modulation across the biomarker range of the disease. With those hurdles in mind, we can step back and define the broader problem of skin immune disease efficacy modeling. One of the goals of modeling atopic dermatitis, and indeed any disease, is to improve the quality of preclinical evidence before clinical investment by using systems that better reflect human tissue and immune biology. In this figure, we have human relevance on the y-axis, and we have biological complexity on the x-axis. The old 2D models for studying barrier marker changes are too isolated and too reductionist in readouts. Cell-free assays are suitable for screening, and mice are not humans, as evidenced by the recent FDA push towards new alternative methods, otherwise known as NAMs. One step up, we have immune cell co-culture systems, which can predict interaction between different immune cells with keratinocytes. 3D reconstructed models that are reproducible for a very narrow marker set, and the same for organoids. And all of this culminates in complexity with organs on a chip. On the other hand, working backwards from humans, we of course have clinical studies which are highly costly, and then we have models induced using human skin explants, which serve the best balance between disease complexity, throughput relevance, and ease of testing. In this context, the model needs to do several things. It needs to induce a relevant phenotype. It needs to quantify that phenotype. It needs to capture donor-to-donor variability. And it needs to respond to interventions in a way that can be interpreted biologically. This commune AD model was developed to address these issues in a human tissue and immune cell context. This commune AD model is an explant-based, non-animal efficacy model designed to interrogate skin immune diseases. It is intended to be flexible as a platform. It can be used with soluble and topical agents, antibodies, small molecules, ASOs, emollients, and other modalities depending on the study question. It can be applied for candidate screening, efficacy testing, preclinical safety work, and mechanistic investigations. One of the important features is that the AD-like phenotype can be induced in healthy human samples while still allowing donor-to-donor variability to be observed and measured. To execute this model, we acquire matched bloods and skins from clinic, process, and co-culture them, and where the timeline allows for multiple therapeutic windows, and efficacy is assessed by a variety of methods. The primary method is through histopathology, through H&E staining, a well-validated and spun-off method from our immunotoxicity assay. This is the classical commune subjective expert grading system. Subepidermal lesions tend to represent Th1 inflammatory insult, as shown here. In grade 1 in the background, we have no damage between the epidermis and the dermis. At grade 2, lesions start to form at this junction. At grade 3, they tend to form together and merge. And at grade 4, you get a full separation of the dermis from the epidermis. For Th2 responses, we tend to see epidermal damage progression. The background remains the same, while for grade 2, we tend to see spongiosis and dyskeratosis start to occur within the epidermis. And within grade 3, we tend to see them merge together and start forming acantholysis. And then by grade 4, you get a full epidermal necrosis where the epidermis disintegrates. And atopic dermatitis in the model tends to only fit around the grade 2 mark. So what you'll see is, as well is because there isn't enough resolution in the data for us to talk about severity of AD, we eventually transitioned to using a quantitative method, which I will show you shortly. The key point here is that expert analysis tends to account for all of these factors assessed by the quantification in the context of the donor skin and whole image. The older data that I will show later will be graded on this scheme, while newer data are quantitatively assessed. So then, to summarize the readouts from the assays, we have quantitative histopathology, including grade damage, epidermal thickness, potentially lesion coverage, but not likely in this model, and immune cell infiltration. For immunohistochemistry, we can have TSLP coverage, filigrin, phosphorylated STAT6, CD1A, CD3, 4, PD1, PDL1 are all detectable within this assay to some degree. And ELISAs by MSD panels such as against IL-5, IL-13, TNF, IL-17, TSLP, TARC, eotaxin, and any of the other ones we mentioned earlier. On the other hand, we can also run FACS on the PBMC fractions that we co-cultured with the skins to detect CD3, CD4, PD1, and IL-4 receptors, for example. On the therapeutic side, the model has been tested with agents such as dupilumab, tralokinumab, lebrikizumab, and is validated against abrocitinib, as well as hydrocortisone and clobetasol topically. The strength of this approach is that we're not relying on one single endpoint. We are looking across tissue damage, immune signaling, and cytokine release, while we are building a cohesive picture. And with that framework in place, we can move into model characterization. In this section, the main questions are straightforward. First, what does the baseline AD model look like? Secondly, how well do relevant standards of care and comparative therapeutics intervene in the model? And third, how does treatment affect timing response? We will move from induction to quantitative scoring to donor background and then to therapeutic response. The first step is defining the core quantitative histology. From the top left, we have autologous PBMCs co-cultured with skin and an AD inducer for the duration of the assay. The PBMCs pre and post culture show strong upregulation of PD1. In the culture supernatant, TOC is strongly upregulated by AD induction. In the H&E images, the baseline epidermis is thin in the background controls, while after AD induction, we see an increase in epidermal thickness. In parallel, we may see increased spongiosis as well as TSLP upregulation. If we look at the histogram distributions on the right, we'll see an increase in positive pixels after AD induction, followed by a shift back after intervention with abro. Together, these measures provide a quantitative basis for assessing whether the model has developed an AD phenotype. What is the AD induction Z-score? For each histological feature, thickness and spongiosis, the Z-score can be calculated as the measured value for a sample minus the mean of the controls divided by the standard deviation of the controls. Then the Z-score that you obtain for thickness or spongiosis can be combined for a total histology score, which allows for the distance between AD induction and healthy controls to be measured for each donor. For example, here in this image, the healthy control has a negative histology score, and the AD has a positive histology score, far higher, and a far higher induction score. Across 26 donors worth of AD induction, we see strong upregulation of the histology score, where values between 2 and 4 are defined as the standard AD induction, and above 4 as strong AD induction. One important observation here is that donor background influences induction strength. Here, we show that donors with an AD background tends to show increased epidermal thickness and spongiosis compared with the total assessed population. In fact, here we have an n equals 4 compared to the total of n equals 26. And when we look at all 4 of these donors, they show elevated AD histology scores and elevated AD induction scores. Ranking them against all 26 donors, one donor here has come in rank 1, one donor has come in rank 2, another one has come in rank 7, and the remaining one has come in at rank 10. That is a biologically important factor. Donor variability is not just noise in this model, because it reflects also the human biology that we are trying to capture. In a simplified system, donor variability can be lost or treated as a problem to be removed. And in this system, instead, it can be measured, ranked, and used to understand how strongly different donors develop an AD-like phenotype. With that, we can ask whether the model responds to clinically relevant interventions. Here, we show data across a dose range for dupilumab and a single dose for abrocitinib. On the left, we have spongiosis, and on the right, we have epidermal thickness. For spongiosis, media controls show a baseline quantification of 20% spongiosis, increasing to 30% on average with AD induction. And with dupilumab, we see a dose-dependent decrease in spongiosis back to close to baseline at around 50 micrograms per mil. Abrocitinib achieves this at an optimized dose. Assessing the epidermal thickness, the media controls show a general thickness of 100 pixels, increasing approximately to 150 pixels, and then decreasing to a minimum at the 10 microgram per mil dose. So in fact, both of these markers, we see differential AD intervention responses versus dose. However, the overall picture is clear. We can see a dose response to dupilumab in this model. And the histology images then shows you how this looks like at a tissue level. We have 3 donors here, row 1, row 2, and row 3. We have media controls on the left, AD induction in the middle, dupilumab off middle, and abrocitinib on the right. So following along the top donor, we've got quite a neat epidermis here with no spongiosis, and that is the baseline thickness. With AD induction, you don't see a thickening, but you see strong induction of spongiosis. With dupilumab treatment, this goes away, and so does it with abro. For donor 2, this is the baseline histology. With induction of AD, it's a bit weaker. You get some thickening and spongiosis, but a bit less than the donor above. And again, this goes away with dupilumab and abro treatment. Same then with donor 3. The neat background histology with spongiosis, and what you see perhaps are rete ridges. So not really representative of thickening here, but it does recover in dupilumab and abrocitinib. This is TSLP staining for the same 3 donors. In the media control, there's not much in the way of DAB signal. With AD induction, TSLP comes up quite strongly. Then with dupilumab, it decreases, and with abro, it's completely abrogated. With donor 2, it's same. You see the same effect, and with donor 3 as well. Lebrikizumab and tralokinumab are both IL-13 targeting therapeutics, so they are mechanistically relevant comparators in AD. Lebri and tralokinumab do not rescue AD phenotype when working with this induction method in our model. Specifically, it also does not work in isolation, but we suspect that when treated together with dupilumab, we could probably reach an efficacious dose of dupilumab much faster. Next, we test abrocitinib at 2 therapeutic windows. In the media control, we see grade 1s for 10 donors. With AD induction, we see an average of grade 2.5 over 10 donors. With abro at the primary therapeutic window, we see a decrease back to grade 1. And when we apply prophylactically as well, abro reduces damage back to grade 1. So this is useful because study design often depends not only on which agent is being tested, but when it is introduced. A prophylactic design and a therapeutic design answers different questions. If an intervention is active across windows, the model can be used to explore both prevention of phenotype development and reversal or reduction of an established phenotype. This histology panel shows this comparison directly. Clobetasol, a dermovate, is a potent topical corticosteroid and therefore a useful topical anti-inflammatory comparator. In this model, topical clobetasol strongly downregulates the AD phenotype. This is important because it supports the use of the model for topical interventions testing as well as systemic or soluble treatments as above. It also provides a familiar positive control style intervention where strong anti-inflammatory activity would be expected. The typical project progression with our model tends to be phase 1, where a pilot of 3 donors are recruited to characterize the AD activity, followed by phase 2, where a 10-donor panel is recruited to test dose response across various test articles. Methods for immune cell collection can include harvesting of the whole white blood cell fraction, including T cells, B cells, NK cells, monocytes, eosinophils, and neutrophils. And readouts are, as we discussed before, the histology AD induction score, a cytokine panel, FACS analysis for the markers of interest, and immunofluorescence for the markers of interest. Pull this together, we can consider the model against a broader NAM criteria. This summary slide brings together the main features of the model. New alternative methods should meet 4 criteria: context of use, biological relevance, technical characterization, and the model should be fit for purpose. In terms of context of use, SCIMMUNE is positioned as a collaborative model where the study design can be shaped around your specific therapeutic question. In terms of human biological relevance, it goes without saying, SCIMMUNE uses unmodified live human skins and bloods and provides an immune-competent tissue context with greater physiological relevance than simpler 2D or 3D systems. In terms of technical characterization, the model has been tested with standards of care, as you've seen, so we are well aware of how to modify the model to suit your needs. The model is also supported by a broader Alcyomics study portfolio in immunotoxicity. In terms of fit for purpose, the value is the combination of the quantitative data, visual tissue data, donor variability, and functional tissue damage readouts. The important point here is that it's not that the model removes biological variability, it's that it allows us to measure biological variability in a controlled human system. I will finish with a brief note on Alcyomics. Alcyomics has a track record of providing preclinical immunotox testing in our model. The company works collaboratively with clients from project conception all the way through to report submission and beyond. And Alcyomics is ISO 9001 accredited. We have been onboarded with major clients, and we are based in Newcastle upon Tyne in the UK. For anyone who wants to discuss whether this model fits a specific program, the contact details are here. Thank you for listening. If you have any questions about the model, specific mechanisms, study design, donor panel, planning, or readout selection, please do get in touch. We will be happy to discuss how the AD model here could be applied to a particular therapeutic question or investigation.